Actin is a highly conserved protein. 95% of actin isoforms share identical amino acids in 66% of their positions. Of the 323 known actin sequences, 312 contain tyrosine at position 53. The remaining 11 sequences contain phenylalanine, a hydrophobic amino acid, instead. Since tyrosine 53 is so conserved in actin, there must be an important structure-function relationship. In one experiment, actin cDNA from Dictyostelium was mutated, and the effects were observed in AX3 cells. Tyrosine 53 was replaced with the individual amino acids phenylalanine, leucine, alanine, glutamic acid, and tryptophan.
The mutated actins were then observed in the presence of DNase, an enzyme that catalyzes the hydrolytic cleavage of phosphodiester linkages in the DNA backbone, thus degrading DNA and reducing function.
Globular actin has a D-loop structure from residues 40 to 50, which is where DNase binds to actin. Wild-type actin with tyrosine 53 stabilizes DNase binding loops and regulates nucleotide binding sites. Due to steric hindrance within the actin structure, DNase is prevented from cleaving DNA and is thus inhibited by actin.
When tyrosine 53 is substituted with alanine, or with glutamic acid, this leads to a reduced affinity for DNase. Thus, there is a higher probability that DNA will be cleaved.
When tyrosine 53 is substituted with tryptophan or leucine, it changes the conformation of the nucleotide binding cleft and ultimately leads to a reduction in function.
When tyrosine 53 is substituted with phenylalanine, this leads to no change in function of actin.
When tyrosine 53 is substituted with phosphorylated tyrosine, the D-loop is partially stabilized by hydrogen bonding between the phosphate group of phosphorylated tyrosine and residues glycine 48, glutamine 49, and lysine 61 of actin. This stabilization reduces the affinity actin has for DNase I, and in turn DNase I is less inhibited by actin. It can now cleave DNA more freely.
In the eukaryotic cell, actin is a highly conserved and abundant protein. Actin is also widely known for its vital role in the function of muscles. It plays an essential role in cellular function, which includes the intracellular movement, cell signaling, cell motility, polarity to the regulation of transcription, and the maintenance of cell shape. From the presentation, you can see that almost any mutation by an amino acid substitution in the actin structure will lead to a change in actin's function.